Hello, everyone, and welcome to this session on genomics. I have the great pleasure of introducing Roberto, but before we get started, I'd just like to quickly go over the Microsoft Code of Conduct for this event. Many of you have probably seen this before if you've gone to other sessions at Build, but I just want to make sure we all take a quick second to skim through it, read over it, make sure we all understand what the Code of Conduct is. In summary, the idea is that we all just want to be nice to each other, create a friendly, professional, welcoming environment where everyone can feel comfortable. All right. So the wonderful person we have joining us today is Roberto. He is a bioinformatician on the Microsoft Genomics team, and he is here to give us an intro to genomics and talk about how you can use Azure to analyze genomic data. Take it away, Roberto.
Thank you so much, Shana. As Shana pointed out, my name is Roberto Lleras. I'm a senior bioinformatics scientist on the Microsoft Genomics team within Microsoft Research. For the next few minutes, we're going to be focusing in on genomics, and particularly how we can fully harness the power of genomics in the 21st century. I'll then give you a brief introduction into how genomics analysis can be done on the Azure cloud using our Cromwell Azure implementation. And lastly, talk about some of the work that we at Microsoft Genomics have been doing to really push the boundaries of genomics and integrate genomics and health data science to be able to put all that data to really drive the field forward. So for those of you who may not remember from Bio 101, we're in world of genomics, we're studying the information encoded within DNA, or a deoxyribonucleic acid. Think of this as our molecular hard drive, and that hard drive contains 3.2 billion letters of information, or bases as they're referred to in DNA. If we were to think about this in the context of a novel, which has on average, standard novel has about 1,300 words per page, it would take 2.5 million pages to encode all of the information that's stored in every single cell of your body in a single novel. That's a lot of information. And so this presents a unique set of challenges for us, and ultimately really demonstrates where the power of the cloud truly lies. It's also really important to remember that this information can really power the research as well as medicine and a whole number of different fields that can benefit from this genomic information that's encoded, whether that be for personalized medicine, identifying infectious agents, or predicting whether or not someone may actually have a possibility of inheriting disease. But dealing with data at such a large scale is a unique challenge that all genomic scientists have to work with. And this challenge really breaks down into two segments. First and foremost, when we sequence a genome, that is reading back the information that's encoded within an individual person's DNA or an individual sample's DNA, we generate over 100 gigabytes of data for that individual sample. In order to be able to do this at any significant volume, to really be able to study populations, we generate enormous amounts of information. So thinking about how we move that data and how we store that data is quintessential. We also have to then consider how we analyze that data. And current algorithms, because of the nature of the data, and we'll talk a bit more about this in particular momentarily, require hundreds of core hours in order to understand that information. And for example, it would take over 9 million core hours of to analyze all the genomic data we would generate if we simply had a full Madison Square Garden worth of samples that we were analyzing. And lastly, we have to think about how we actually integrate and structure this data in a manner that isn't just simply focusing on the genomics information itself, but how we interpret it and how we add other pieces of information that can be valuable into that particular data set. So let's talk a little bit about what a typical genomics analysis pipeline looks like. We first start off with taking tiny pieces of DNA, and those are interpreted through a number of different technologies that ultimately generate tiny pieces of information of that 3.2 billion base pair DNA sequence that represents each the genome of your cells. So first and foremost, we have to move hundreds of gigs of information per sample that are coded in files that are called FASTQ or BAM files. And we have to then consider how we can actually add some structure to make sense of all the random pieces of data that are there. And I mentioned here that this is what we call in computer science world the NP-hard problem. And if you're not familiar with that term, it's just a fancy term in computer science. It means that something's extremely difficult to do in order to be able to run. Once we have this order put on the information, we then have to then interpret for individual samples and across in pieces across the individual sample where there might be similarities or differences from what you would normally expect in that DNA sequence to be. And we call this in the genomics world variant calling and filtration. And currently this field already utilizes machine learning models to accurately spot changes in the DNA sequence or mutations. We then have to think about how that information actually impacts what we're looking at in terms of the overall genome and the genomic information that's encoded. But in order to be able to do this, we actually have to then take insight from a number of other pieces of information, whether that be data that we obtain from the clinic, reference databases that tell us whether or not a particular mutation may be advantageous or disadvantageous, image data, a whole other pieces of data. Working on Azure, we have the benefit of being able to leverage other existing tools such as the Fire APIs to be able to bring that data into the cloud in a secure manner, structure it with Azure products and services, and then ultimately we have to finally be able to present that data back and use it for different individuals who may be interested in learning what that information encodes, whether that be our researcher, a physician, a patient, or anyone else that may be interested in looking at that data. From a computer science perspective, all of these different steps encompass a number of different research and exploration avenues. And so I encourage those of you who may or may not have actually thought about doing genomics research within the context of a broader computer science background to really think about it, because we have to understand the data as well as, since this is probably the most precious piece of information one could actually have and look at, we have to make sure that that data is being protected and that the data can actually be interpreted and utilized, which encompasses a number of different fields. Moreover, the different fields and the different tools that people are used to interacting with that data, we have the benefit here at Microsoft of having all of those available at our disposal. But ultimately, we have to be able to figure out and properly pair the right toolkits and the right tools with the right steps and the appropriate users for that. And so within Microsoft Genomics, it's fundamentally our goal and mission to be able to try and make sense of this process and to be able to streamline and automate this process as much as possible to enable our users and researchers, physicians, and scientists to make the most use of this information. So let's now dive into a bit of some of the individual work that we've been doing. And our initial focus will be in understanding how pieces of DNA ultimately are related to one another. So if we took these old things called newspapers, and I asked you to tell me what happened on June 27th, 2000, but I decided to make it more difficult and blew those newspapers up before I handed you the remains and asked you to tell me the information. We as computer scientists know that we can take the random pieces of information, those short snippets that would remain after the explosion, and use our problem-solving skills to start piecing back together the information that's encoded. And just as a primer for those of you who may or may not be aware, June 27th, 2000, actually is known as DNA Day, the day that the human genome was first published. This actually represents a huge computational challenge, and what we're doing every time we analyze an individual sample of DNA sequence data. We look at between tens to hundreds to even thousands or millions of copies of DNA from a sample that are all randomly fragmented because of the fact that DNA sequencing instruments don't have the ability to read all 3.2 billion bases at once. So we have a random assortment of pieces of genome represented in our data set. About 2.2 to 3 billion reads of that information. We then perform our overlapping step and use other bioinformatics tools to be able to reassemble this information, to recapitulate the actual reference sequence that we're interested in looking at to identify potential changes that may exist. Ultimately, the more data we have, the more chances are that we have enough pieces that overlap with one another to be able to recapitulate the sequence. And so thus the resultant reference is of higher quality. But the problem is that in order to be able to do this, this requires a huge amount of computing power and storage capacity. This process in bioinformatics is commonly referred to secondary analysis, and it's ultimately taking unstructured genomic data and adding some level of structure and understanding to that to be able to identify what may be happening in an individual sample. And one of the best practice pipelines that's currently been implemented by the Broad Institute of MIT actually performs this analysis. It performs an initial overlapping alignment step, a number of quality control steps, and then fundamentally identifies our mutations. The current average compute time for this is approximately 30 hours. As part of a collaboration with the Broad, our Microsoft Genomics team has actually optimized this service on Azure. And we can take an average and significantly reduce the average compute time for a sample to be processed. And so this service ultimately is currently available, but it does one specific pipeline. And the world of genomics is rapidly evolving. First and foremost, if we look over the last 15 years or so, those of you in the familiar with computer science will be familiar with the term Moore's Law. Well, genomics is the one field out there that actually breaks Moore's Law. And the cost of human genome sequencing, thanks to an explosion of new technologies, has actually resulted in the cost of a genome rapidly depreciating over the course of the last 15 years, and currently sits between $100 to $300, depending on what technology you're using. Moreover, this field is constantly evolving and adding new layers and interesting pieces of information. Innovations such as long-read sequencing, which allow us to look at bigger pieces of DNA, single-cell sequencing that allow us to actually use molecular tags to keep track of each individual cell in a sample, and spatial sequencing, which actually allows us to keep a location information actually within a tissue to be able to understand where DNA and sequence is actually coming from. So these other pieces of information, in combination with rapidly lowering costs in sequencing, mean that as a team, our goal is not only to simply enable specific pipelines, but rather our focus is shifting to actually building a rapidly scalable platform that's elastic to allow for the continued innovations in research, while enabling, once you've identified those insights, be able to actually deploy those at scale to actually benefit the human population. And the first component of that is implementing a workflow engine that was developed at the Broad Institute called Cromwell. Cromwell ultimately has the ability for us, enables us to be able to run any scientific workflow that is containerized using and leverage Azure Batch resources to provision individual virtual machines for a given task to process that data. And so we have authenticated access, and users can either supply data directly using REST APIs, or they can use Azure Active Directory services to actually be able to upload data to blob storage and ultimately initiate and automate analyses and ultimately have that output be reported back to them. Here's a brief demonstration on how Cromwell in Azure actually works.
Is there audio with this video?
Apologies for that. So this video is actually embedded within the shared materials. So since there is audio with it, I would encourage anyone who is interested to go back and listen to that audio. Apologies for the technical difficulty there. But what we'll do is that I'll move forward. I'll move forward for sake of time. As I encourage everyone to come back to this momentarily. And so fundamentally, what we're looking to enable, utilizing this Cromwell on Azure pipeline, is not only to be able to automate genomics analyses and be able to plug and play specific secondary analyses, for example, like the Microsoft Genomics service on Cromwell and Azure, but also be able to facilitate the transition and the preparation of data into what we call tertiary analysis to enable us to actually drive machine learning and data science analyses of genomic data, integrating that with other pieces of information that a user may have, and then fundamentally present that with Power Platform services. So to this end, this is an example of a polygenic risk score prediction dashboard that we've developed in collaboration with a customer, where we're actually, and this is actually a real-time dashboard, and I would encourage anyone who looks through the materials after to really actually dive in. This is interactive and can be examined. But actually, take a look at 4 different machine learning algorithms that have been trained on a number of different SNPs based on their prevalence and their the statistical evaluation in an individual set of samples to understand a population that has been sequenced. Why is this important? Because fundamentally, what we're looking to do is, it's not simply just research. We're actually looking to empower healthcare and empower physicians to be able to actually make a difference on the Microsoft utilizing the platform that we're building. And so since the audio seems to not be streaming properly over the Teams app, I will skip playing this video, but I would encourage you to a story about one of the collaborations that we currently have with Seattle Children's Hospital, looking at building a model for predicting sudden infant death syndrome, or SIDS. So with that, I'd like to open the floor to any questions that users may have. And thank you all for attending.
Thanks so much, Roberto. I'm going to send your video to the audience so they can see you while you are answering some awesome questions we've got. So I wanted to start with some questions just about you and your background and genomics at Microsoft, which is not maybe what most people know Microsoft for. Could you start by maybe answering the question, what exactly is a bioinformatician and what do you do at Microsoft?
Certainly. As Shauna pointed out, said I am not actually a computer programmer traditionally by training. I am a bioinformatician, and so my background is actually I've spent just as much time working in a lab with leeches and with C. elegans, which is a small semi-microscopic worm, and with human head and neck cancer samples as I have programming. And fundamentally, bioinformatics is the fusion of computer science and biology. And so I've spent time both on a bench actually learning molecular techniques, as well as I also have some traditional computer science training as well. So the bioinformatics, as I said, is a really interesting, as I said, field, and still one that's very much in high demand. So if you have an interest in biology, as well as you're attending builds, so you have some interest in computer science as well, bioinformatics is a great field where you can actually toe the line between, and ultimately you're enabling researchers to understand data and biological data using computer science and programming techniques.
Awesome, thank you. What a good explanation. I'm curious, what kinds of jobs are there out there for people who maybe also don't have a traditional computer science background, or maybe even for people who do, but are really interested in the sciences like biology or chemistry or physics, all those great life sciences?
It's an excellent question. So fundamentally, all of these what we called hard science and soft science tech fields generate and continue to generate lots and lots of data, whether that is genetic data or information about metabolites and chemical data and such. And so fundamentally, there's a huge demand for individuals who can and are apt and capable of working with large amounts of information on a computer. And so in order to get into bioinformatics or the informatics field in general, you're definitely you're going to want a strong computer science background. Because you'll fundamentally be working a lot on the command line and also working with high performance computing and the cloud and such to do that. But you also will want some understanding of the basic science field that you're interested in. So in my case, I have a lot of background in genetics and biology and biomedical science. But there are bioinformaticians that are really well focused in protein biochemistry, which admittedly is completely beyond my expertise, but to have similar big data challenges as well. And so you want to say you have to start with your computer science core, but then you can start, and collaborate with the basic science field of your interest. And that's really how you can then transition pretty easily into this field.
Awesome. Thank you. That's great. We have just a ton of questions coming in. I wanted to ask a couple. We had some questions at the beginning about different programming languages in bioinformatics. So one of the questions was, what is the preferred programming language for pursuing bioinformatics?
So this is a age-old question and has rapidly evolved even over the course of my career, which it's not that long. Currently Python is the dominant language in the field. If you're more interested in the really hardware acceleration and more focused more on the computer theory aspects of things. A lot of, I said, that world really works in C# and in some of the more machine-focused languages. I said in that regard, I have friends and collaborators that are F# programmers, said, for example. And then if you're looking to more manipulate data and really be able to interact with data and particularly interrogate and understand it, I said Python is always a good go-to, but there also is a very rich development for R. And so being an R programmer you can have a very successful career in terms of understanding data, working with R. There's a few other languages that are really kind of up and coming. Rust is one that really seems to be starting to take off within the bioinformatics world. And then it's always handy to have some Perl or general batch scripting knowledge as well, because you're always working with different environments.
Awesome. Okay, here's a question more from the biology side of things. Someone was curious, why is it required to fragment the genome before you kind of start this process of analyzing that data?
That's an excellent question. So we're dealing with in each individual cell, 3.2 billion bases. And those bases are only in, are the only thing that's really structures those are denotes pieces of them are the individual chromosomes that are in your genome. So you have 23 pairs of chromosomes in a human genome. So and each of the chromosomes varies in size. Unfortunately, from a technology perspective, we just don't have the ability to actually read an entire genome at once. Moreover, molecular techniques make it extremely difficult for actually keeping all that DNA, those DNA solidly in one piece together. So you have two challenges, both from the molecular side, biology in terms of keeping the DNA intact, particularly since you're generally harvesting DNA from, and ultimately you have to kill whatever you're harvesting the DNA from. And the moment you kill something, apoptotic processes introduce enzymes that will actually start cutting DNA. And then you have the technological challenge of actually being able to read that data. So we just simply haven't bridged that challenge. We're making strides there with long read sequencing and technologies in that regard, but we still can only look at things that are kilobases to megabases. So hundreds of KB to megabases in length is still kind of the upper barrier to that. So that's
awesome.
why.
Thank you. I'm going to do one more quick question, and then I think we'll switch to our last slide. One that just came in: Linux or Windows for genomics? Which is the better platform for genomics?
I am actually Linux trained first and foremost, and actually kind of my go-to. Sorry Windows, but the fact that you can have Linux embedded now within your PC is pretty awesome in that regard. But yeah, most tools are kind of generally developed with Linux OS in the background in mind. So,
yeah,
so
I will say,
let's go.
I'll say Windows subsystem for Linux for the win. Now we can have it all.
Yes,
awesome.
Absolutely.
Okay. I'm going to switch us over to our very last resources slide. If you could flip to that one. Fantastic. Okay, thank you everyone for coming. Thanks for all the amazing questions. We're so sorry we couldn't get to all of them. There's just so many coming in. I will say, if you enjoyed this session, please do go ahead and fill out the survey for this session. You can find that at aka.ms/studentzone2020 survey. And the session code for this one is COM215. These slides and other resources will be available for everyone at aka.ms/studentsatbuild. Thanks again for coming. We really appreciated all your fantastic questions. Here's one more slide. I put these in the Q&A as well at the very beginning. So these are some great links to learn more about genomics too. Thanks, everyone.